Today I'll be talking about my own publications. My own publication that recently came out that I was participating as one member of many scientists, and it's a fairly controversial topic. Heads up, because it talks about how the synthetic pseudouridines that were used in the construction of mRNA vaccines might be involved in promoting cancer growth, not cancer development. So this is, we're not saying that these mRNA vaccines are causing cancer, but that they might be promoting cancer growth. And this might be specifically be happening primarily due to suppression of the innate immune system. So that's what we're going to be talking about based on that review. And why we're doing this is primarily this group of scientists that I'm very privileged to have become part of, is to find solutions to the problems. We're not here to necessarily point fingers and blame anyone, because at this point, if there's a problem, we're more interested in figuring out, if there's a problem, how do we solve that problem, okay? So that's what really talking about. And basically, what are these pseudouridines? These are in the mRNA vaccines. Let me introduce myself. My name is Dr. Raszek of Genomics, okay? Let's get going. The big topic is, of course, mRNA vaccines. When they're made, they had this not typically natural component in them. So they are called N1 methylated pseudouridines. Basically, think of it this way, but I'm gonna refer to them as synthetic pseudouridines from now on, because they're not typically seen in nature at all. When you have RNA is made up of 4 chemicals, A, C, G, and U. U stands for uridine. And it's basically an arrangement of these 4 chemicals in a specific pattern that is the genetic information that acts as a blueprint that tells cellular machinery how to use mRNA in order to produce a protein of interest, okay? So in this case, this would be the spike protein. So all of the uridines in the genetic code of mRNA used for vaccines were substituted, and those synthetic pseudouridines were used 100%. And there's a very specific reason why this was done. But we're saying that might be a double-edged sword. So why mRNA vaccines? Why this was the technology, the primary technology that we use in order to respond to the pandemic? And the reason why is because this technology has some amazing benefits that you have to understand. One of them is the fact that it can be very rapidly developed and executed super quick. So this allows some of the fastest response time when it comes to some biological threats, such as a pandemic. That was the number one attractive component, that why that technology was used, precisely because it allowed some of the fastest development of vaccines. And number one. Number two, it also allows very rapid change from one type of vaccine to another. So we can very quickly change the variant used in the vaccine. And that's exactly what we did during the pandemic. We were switching from one type of variant to another throughout the pandemic. It took us a while, but eventually there were switches that were observed, right? And that's another benefit of this technology. But mRNA technology was never used before in humans, the first time ever. So when we employed it so rapidly, we also employed it with some gaps of knowledge. We simply never had enough time to test what the consequences of what we're doing might be. Why? Is because we employed every single trick in a book to achieve one singular point, and that was to produce as many antibodies in a vaccinated individual as possible against spike protein. That was the number one goal, because this was what was desired the most, to ensure that these vaccines are working properly. And basically, I better double check my mic. Yeah, good. And basically, this might be the double-edged sword, because we used every single trick known to science in order to increase the amount of antibodies, and that included also suppressing the innate immunity as well in the context of these mRNA vaccines. And I'll get to it basically right now. So why mRNA vaccines? Because they had 2 major benefits. First, one big problem was to solve how mRNA should be delivered to your cells. So this amazing lipid nanoparticle construct was created that finally allowed to achieve that problem, solve that problem, and achieve an easy entry of these genetic mRNA content into our cells. But the other one, that was super big one, super important one, was the use of these synthetic pseudouridines. And why is that? Because it achieved three big things. Number one is it suppressed that innate immunity. Why would we want that? We wanted that because previously immune system was recognizing it powerfully and destroying it. So by suppressing the innate immunity, it guaranteed increased longevity of basically the genetic construct. And if you increase its longevity, it means you can increase how much more spike protein is being produced. And if you're increasing more spike protein being produced, the outcome should be you're gonna produce more antibodies. Whether that's actually true or not, I think that would be the reasoning here. All right, so that's big deal. It basically allowed the construct to hide itself away from recognition of the immune system. So that's the biggest benefit in terms of how this genetic material could survive longer. The second one is that it increases the stability of that construct as well. So in its own accord, it makes the actual molecule more stable, and it also lasts longer because of that. And the last one, the one that is also very welcome and very good one, it was the fact that use of these synthetic pseudouridines in some of the studies showed that they reduce cytotoxicity. So that's very welcome. It basically means that it decreased how likely some cells were going to be destroyed because of encountering such a product. So of course you want minimal cytotoxicity, you want minimal destruction to your cells that encounter this. And so that was very welcome, right? So this is all great, and this is one of the reasons why the mRNA vaccines were heralded as some of the greatest scientific achievement ever, because of this. But it's this innate immunity that might become a potential problem, the suppression of that innate immunity. So now let's start breaking this down, okay? So what are we talking about? So in order for me to explain specifically, we need to start with what is referred to as pathogen recognition receptors. So we have these receptors as part of our immunological system, including innate immunity, are these receptors that recognize everything under a sun that is either foreign or abnormal, meaning our normal cells are no longer looking normal, such as, say, cancer cells. So there's changes in the makeup are happening, and of course those changes are leading to three-dimensional changes. We can recognize that with these receptors, recognition receptors. So we can recognize RNA, we can recognize DNA, we can recognize protein. All of these have very complex three-dimensional structures, but also sugar molecules can adopt very complex three-dimensional structures, and any of that also can sugar molecules and proteins can also interact with lipids, which are fatty molecules that are also can have their own shape as well. All of this can be recognized by these receptors. These receptors are inside the cell as well as outside the cell. So inside the cell, it can recognize if the cell is already invaded. It can signal that information. Of course, if the problem is moving outside the cell, that can be recognized. So these are very good normally. Sometimes they can be abused. And how the vaccines, mRNA vaccines with the synthetic pseudouridine, trick the system, because what normally happens, let me back up. What normally happens is these receptors then send a signal from the surface. When they recognize the problem, they send a signal to the nucleus. Now, nucleus is like your brain of the cell. This is where you house all the genetic material. And basically, its information tells the cell how to respond. And you do that by sending a bunch of mRNAs from the nucleus, and basically that determines how the cell will respond. One of those responses is production of a molecule called interferon. This molecule is a big part of the innate immune system, big one. It's one of the big molecules in inducing inflammation. Inflammation is good when it fights a problem. Inflammation is bad if it's destroying or attacking your own body, right? So that's when you don't want it. But otherwise, you can see how interfere. We'll see how interferon is important in basically making sure you're protected. How does it do that? It can interact with certain type of cells, immune cells called dendritic cells. These type of cells, they can do different things, but one of them is, for example, they can present antigen. They're antigen presenting cells. Antigen is a fragment of a problem that was discovered. So that's what antigen means. It can present that fragment of either something that invaded us or cancer cell to the immune system, so they can start training the immune system. Hey, this is a problem. You need to start recognizing this, dealing with this. And they also play a role in helping to activate T cells. T cells are killer cells, ultimately to get rid of a problem. In the very end, you need to kill all of the damaged cells or cells that are a problem. And you need killer cells. There's two types of these killer cells. There are either T cells, or at least two types that I'm going to mention, and natural killer cells. Those are the only two types I'm aware of. I wouldn't be surprised if there's more. Eventually we'll discover something else. But these killer cells basically will clean up things. So activation of T cells can also be called cytotoxic T cells. They basically, that's what we're going to be clean up, and this is what is important to fight your cancer. You want to destroy cancer cells with these killer cells. Okay, so you can see now how potentially when you remove interferon, that could be a problem down the road. And this was, but unfortunately, this wasn't demonstrated with the mRNA vaccines that were using this synthetic pseudouridines. This was only up. We discussed this paper that only came out in 2022, which showed how different mRNA vaccines might be influencing the interferon, and how that in itself might be influencing cancer growth. And so this is like the seminal paper that we focus big time in this review, and I'm going to tell you about it. So basically what the authors of that paper did, they used mRNA vaccines that were either totally 100% natural, or they were 100% had that synthetic uridine, okay? And they injected mice with these vaccines. They were not for spike protein, they were for something else that was on the surface of cancer cells that they were also injecting these mice with. So these were vaccine against cancer, a specific type of cancer, but the same mRNA technology, and they use 100% natural mRNA vaccines, or 100% synthetic pseudouridines vaccines. And what they noticed is that one thing they noticed is that there was a drop in the interferon production, kind of like what you expect, right, from these vaccines, because that's what allows them to last longer. But when they co-injected the mice with those specific cancer cells, the mice that were immunized or injected with the mRNAs that had 100% synthetic pseudouridines, their cancer cells were capable of growing. Not only was there drop in that interferon production, so suppression of innate immune system, but that seemed to correlate with the growth of those cancer cells that these mice were co-injected with. Okay, they were also injected with that, versus 100% natural mRNA didn't seem to have the same level of progression of cancer. So that's big deal, obviously, right? But they did a lot more. So the next thing that they looked at is, we're going now talk about programmed death 1 molecule. And basically this molecule is, and if it sounds ominous and foreboding, it's because it is. It's a molecule that can come up on the surface of T cells, and it acts like a switch that eventually allows this molecular environment to switch off that T cell from being able to do protective function against something like fighting cancer. So having this programmed death 1, PD1 molecule on the surface of T cells is not a good sign. Indeed, it is foreboding and ominous. And what the authors noticed in that publication is that, again, mice that were injected with 100% synthetic pseudouridines, they had higher levels of these type of T cells with PD1 molecule, which signifies exhaustion of these T cells, than vaccines that were 100% natural. Okay, so, and that again correlated with the cancer growth. So speaking of PD-1, that's also the molecule that is exactly targeted by immune checkpoint inhibitors, the new anti-cancer medication, which is made out of IgG4. It was attacking that very molecules to block it so that you could no longer use that switch, and that would allow the T cells to last longer in order to protect you from cancer. Hence, this is an anti-cancer medication, and indeed it worked fabulously in some. But because we use IgG4 antibody, we discovered that IgG4 antibody in a bad cancer microenvironment can induce turbo cancers, and I made a video on that, so check it out. This is now a video series that I'm producing on the topic. So and it's specifically, it's the stock of the IgG4 antibody that is the problem here, and how that it can promote turbo cancer development. So that's how you can see all these connections in my studies, right? So that's the medication that targets the PD-1. But if you have too many T cells with it on it, then other immune cells or cancer cells could actually trigger that and take out those T cells. So we don't want that in our cancer environment. And the last thing that I'll mention from that paper, I'll talk about macrophages. So macrophages are very super cool immune molecules that if you think of what is their role, their primary role is to basically take care of you, really to make sure that your body is doing well by making sure that it's protected and it can swallow problems such as pathogens. You can literally just swallow them whole and then break them out. But it also can be involved in making sure that everything is working properly, is fixed properly, and so on. So macrophages are very good, but we can subdivide them into like two groups, and depending on which group they belong to, one group can be more beneficial for cancer fighting or be detrimental for cancer fighting, but it could be beneficial somewhere else for the other group. So we call them M1 macrophages and M2 macrophages. And M1 macrophages are the good guys for cancer fighting. That's the guys you want in the cancer to destroy cancer. Why? Because they release a bunch of chemicals, and some of these are reactive oxygen or reactive nitrogen species, and those are toxic chemicals. They're super destructive. So you want them when you fight cancer. You don't want them anywhere else to attack your own body, but for sure when you're fighting pathogens or cancer, you want them. But they also release a bunch of cytokines that specifically change the microenvironment to basically induce the fight against the cancer, and some of that involves in the end inducing bringing T cells, those killer T cells, to the environment, literally like sending a signal, come here, and helping them get activated into that role to do this. So really, really important function. Okay, now then there is M2 macrophages, and those are the bad guys when it comes to cancer. So what they do is they are called anti-inflammatory. They release different types of cytokines that now reduces inflammation in the environment. Inflammation in this context, think of the inflammation as a marker that there is a battle going on and your immune system is destroying something. So inflammation in a context when something that is attacking is being destroyed, that's good. But if it's immune system attacking you, inflammation is not good. So depending on the context, inflammation can be the good or can be very bad. You don't want to have inflammation in your body when you're not under attack, because basically it runs the risk that your own body is under attack or could be attacked. Right, so then M2 macrophages induce that anti-inflammatory environment, and that anti-inflammatory environment includes interleukin-10. This is something that can be used to also reduce the number of those killer T cells as well. And interleukin-10, also remember, I brought up in my video series dedicated to IgG4 that we are seeing IgG4 produced against spike protein in the vast majority of the mRNA vaccinated individuals, right? And one of the papers so far, only one suggested that I know of, suggested mechanism behind that, and it is the interleukin-10 itself that is promoting this class switch observed in mRNA vaccinated to start producing these IgG4 antibodies. So, and also M2 macrophages promote the angiogenesis. That basically means development of new blood vessels and basically blood network. And of course, you can imagine for cancers, that's absolutely desirable. That's exactly what cancers would want, because if they want to continue growing in size, they need basically oxygen and blood supply, absolutely needed, right? So you can see how, again, this might be so beneficial to cancer cells to have M2 macrophages in their macroenvironment than not M1. Now let's bang back to turbo cancers again, because in the video series that I discussed, I talked about how the IgG4 stalk, what it does, it helps the cancer microenvironment by inducing this state towards the M2-like macrophages in the cancer microenvironment. So you can see how there's now double whammy interplay potentially going on here because of these synthetic pseudouridines, because you now have induction towards M2 macrophages, induction towards IgG4 production, while you suppress innate immunity that might be reducing access of killer T cells to that cancer environment. So that's basically how all of this is linked together. But M1 macrophages, another thing, what they do is they're so powerful that they also will reduce likelihood of cancer metastasis. So when you remove them from the environment, you're more likely to start metastasizing as well. And in these experiments on mice that these authors I was talking about did, they also noticed that when the interferons were reduced in these mice as a consequence of using mRNA vaccines that were 100% synthetic pseudouridine, and how that increased the level of those T cells that with the PD-1 on top of it. Well, when that all happened, these type of cancers, not only did they grow, they also had an increased likelihood of metastasizing as well. So you can see how this is now all connected, and how innate immunity might be playing big role as well. So that's the innate suppression of the innate immunity, which was attempted in order to make sure that the vaccine lasts a very long time, so that we can produce great antibody response, might be causing other problems as well. We also talked about two more that I'm quickly gonna summarize for you as well. And one of them was the fact that these pseudouridines that has been reported once so far, that it might cause frame shifting. And so then what is frame shifting? Then basically, remember, mRNA is a blueprint to produce a specific protein of interest, and a protein you can think of as like a robot. But in order to do this, in order to translate the genetic blueprint into production of a protein, you need a machinery jump on the mRNA and start decoding the genetic code, that which tells how the protein is built little by little. And these synthetic pseudouridines can make that robotic machinery that is responding to the genetic code accidentally slip and jump ahead. And now it no longer reads correct genetic code. And as a consequence, it can start creating rubberish products, something that we don't really even know what it might be, but it wouldn't be spike protein. So, and in that publication, apparently antibodies against such abnormal products have been discovered. So these would be called neoantigens, these new abnormal products that we don't know what necessarily they're doing and what they would be looking like. They would be called neoantigens, right? And what is the reason why we brought it up in this review as well is because it's now known that certain cancers also can cause frame shifting and utilize frame shifting in order to produce these abnormal neoantigens, so fragments of some abnormal proteins. And they do this to technically confuse the immune system response, and it's a way of protecting them. One possibility how they might be protecting themselves is what is believed that cancer cells might be presenting specific antigen specifically too much in order to induce that IgG4 class switch, which will then come in handy to start cloaking the cancer environment from being recognized by the immune system. So, and therefore frame shifting perhaps could be one of those ways that will allow to produce a lot of antigen in order to provoke our own immune system to start switching towards IgG4s. But you know, all of this is like, well, is M2 macrophages always bad and IgG4 antibodies always bad? Well, no, you can imagine that it might be in circumstances where the same thing that can help cancer grow in another context could be extremely helpful. And when I think of, say, angiogenesis and anything that can promote, for example, blood vessels growth, this is when I think of like tissue regeneration. And all of these mechanisms that might help cancer might also be extremely useful in something like helping repair in our body. So it's not that it's all bad. There is a purpose why it exists. Is that cancer cells have found a way to misuse or abuse some of the complex systems that are available to us in our body to survive and grow and become basically a own entity that wants to survive on its own and basically use our own resources to live. That's what cancer would be. So basically that gives you another option, is that maybe that's number 2, right? And the last one that we mentioned is IgG4 antibodies, but we mentioned that we're going to have another review that talks specifically how IgG4 antibodies could be involved on their own in cancer development, and how could the synthetic pseudouridine be involved in production of IgG4s. As I already told you, one possibility is because they could promote those macrophages to start creating microenvironment that can induce development of IgG class switch, right? That's one option. Maybe frame shifting could be another one by producing too much specific antigen. But remember, I also mentioned that when these synthetic pseudouridines were introduced in these mRNAs, one of the presumed benefits was that the genetic material can last much longer, can be then used for producing spike protein longer. You can produce more spike protein, and then you'll have better antibody response that way. Well, obviously it means producing more spike protein. Perhaps we're producing too much, and then it's leading to presentation of the spike protein way too much to the immune system, that in its own right is now also triggering the switch towards IgG4s, and we'll talk about that in future videos. And I've already started that basically when I'm starting to discuss the turbo cancer videos, so check those out as well. Yeah, basically this is it. That's the major components of the review of how synthetic pseudouridines could be a problem when it comes to developing cancer. Now, what is the solution? Well, we already mentioned, right, that one of what the authors of that paper did, they were using 100% synthetic pseudouridine mRNAs versus some that were completely natural. So what we're calling for is stop using, at the moment, completely stop using mRNA vaccines that have 100% pseudouridines. We promote the concept of precautionary principle. Until we know better, we should no longer be using, while we rapidly figure out what should be the appropriate content of pseudouridines that might not be triggering this, because that very well might be the case. Or we very well perhaps can get away with completely normal mRNA, and none of this will be triggered because enough antigen could be presented that there will be the same immunological response we want, but not all of the potential negative side effects that we're witnessing because of incorporation of these pseudouridines. So that should be investigated. And for frame shifting was caused by these pseudouridines, by allowing it to jump the machinery that's supposed to use the mRNA genetic blueprint by jumping forward, but it's in specific places. So by removing it from that place and just simply using uridine, that could fix the frame shifting problem as well. So clearly there are easy solutions available, and we're saying we need to start looking at this because potentially this is an easy fix moving forward, and we can test this, we can figure that out. And another thing we're mentioning we should be doing is we should start looking at how much antigen, meaning say spike protein, produce, or any antigen we use post-vaccination, how much is enough to properly induce the immune system without inducing any of the problems we're proposing might be happening by having 100% synthetic pseudouridines in mRNAs. Perhaps we only need much lower quantity, perhaps not much lower, but clearly not 100%, but we need to figure that out. And then we need to figure out what is the right content, or perhaps none at all, to produce enough spike protein to induce immune system reaction. We want those antibodies, but not trigger other problems by creating microenvironments that could be promoting cancer growth. So that's basically the take-home message of this review. So again, not here to point the fingers. We already know that more and more science is emerging for potential issues when it comes to mRNA vaccines. We're much more interested in figuring out, okay, what is the solution to make sure that we no longer see such problems in the future. If it's even a problem, we need to confirm this. This is all preliminary hypothesis that how potentially the makeup of mRNA vaccines could be problematic in cancer environment. That's what that paper was about. Whew, all right. And I didn't get through everything either. So basically, that's it. Check it out, please. However, I gotta warn you, if you do check out the paper, that there's some graphic images inside of mice post dissection to show specific type of cancer growth. So you need to, warning that that this might not be for everyone. If you choose to download this paper, get this paper and read it, but definitely check it out. Let me know what you think. Please leave us a comment. Please subscribe to the channel. If you haven't already, please share this video. This is how we grow. Clearly, this is a great privilege to be able to actually produce certain theoretical thinking, scientific reviews, and create videos on it as well. So that's part of that is obviously thanks to your support. Why? Because this group of very intelligent people, they came across me because of the videos, right? And I've actually made videos on their prior literature, and this is how I got invited to basically participate in this collective pool of certain wisdom development. And I got to tell you, these are smart guys. I learned a lot, and I'm very, very happy that I have such at the moment intellectually positive influences. So thank you for your support. And last thing, also please check out the Patreon account where we post additional content that does not make it to YouTube. And finally, never forget to get out to nature and enjoy Mother Nature as much as possible. Something tells me that is good for your M1 macrophages and your T cells. So this is a good way to basically perhaps stimulate your immune system to the best shape possible. And I'll see you next time. Bye.